Okay, can you hear me? Okay. Thanks.
Nope, you're good. I'm going to put your slides up.
Okay. Thanks for having me here today. I'm currently a professional researcher at UCSF. I used to be a postdoc there, and I'm a computer science. I work in the neurology department, but I'm a computer science scientist. And today I'm going to talk a little bit about my work on using computer vision and other imaging techniques for analysis of a whole human brain digital pathology. And this is actually, some of this information is in one of my preprints online. So if you guys want, you can check it out later. And so what I do, what I am, what are you doing? I'm in a neuropathology and neuroscience lab at UCSF. And we're mainly interested in studying Alzheimer's disease and other neurodegenerative diseases. And why do we have this focus? Basically because, especially in Alzheimer's, it's the only main cause of death in the US that is going up instead of going down. Other causes like stroke, HIV, heart disease are going down. And we're still not doing a good job in improving Alzheimer's. And this is actually a huge burden for the health system. We estimate that by 2050, we're going to have 1/8 of the population with Alzheimer's disease, and that's very costly. And it's also a big social burden, big burden for the caregivers and family. But despite all this research and investment in research that we have, clinical trials, we still don't have a really effective treatment for Alzheimer's disease. And why is that? And why does it have to do, what imaging has to do with it? I'm going to explain. So one of the main problems we believe with Alzheimer's disease is that the diagnosis comes when it's too late. We know nowadays that Alzheimer's disease, the disease begins affecting the brain at least 10 years before the symptoms. So probably what's going on is that when you get a diagnosis and begin some treatments, your brain is sustaining a lot of damage. And problem is, why don't we do a screening and try to get an early diagnosis? Well, because we don't have a good way of doing this early diagnosis. We still don't have reliable biomarkers. We still don't have reliable neuroimaging. And that's where this project comes in. We want to improve, try to improve the Alzheimer's disease early diagnosis biomarkers using imaging. And we're targeting one protein called tau. So this protein is one of the proteins that you see that builds up in the brain when someone has Alzheimer's disease. You have two main proteins. One's called beta amyloid and the other is tau. And tau, but tau is interesting because it has a really good correlation with clinical decline in Alzheimer's disease. So what happens basically is when you develop the disease, you're going to have this protein. That's why you begin having symptoms, basically because the neurons are dying. When you have this protein spread around other limbic areas of the brain, like the hippocampus, is when you begin having some mild symptoms like memory loss. But by the time usually when patient gets the diagnosis, you're going to have this protein all over the brain. And that's when you have like sustained brain damage and cell death and like the severe symptoms. So what if you could image that protein? And just like to let you know, the only way nowadays, the only way to really have a real final diagnosis of Alzheimer's is using pathology. But when you have pathology, it's already too late. So what if you could have a way to image this protein early and use this as a screening and to begin early treatment, some kind of early therapy? That's our goal. And actually, you have some PET tracers. Do you guys know PET scans? What's a PET scan? So a lot of pharmaceutical companies and research labs are developing PET tracers for tau. There's quite a few of them nowadays in the market. But problem is, we know they're not reliable. we know there's a lot of off-target signal, and that's why you still can just use one tracer and go to the doctor, or maybe I have some Alzheimer's case in my family. I want to know if I'm developing this. A patient cannot just go to the doctor nowadays and have PET scan and have a definite diagnosis. You do see papers, if you check the literature, where you're going to have big cohorts where you can do classification and see this difference. The problem is that you still don't have a biomarker that you can do like diagnosis at the individual level. So how do we improve these PET tracers? We have to validate them. You have to know how they behave in the presence of tau protein in the brain. But how we do this in vivo, I mean, when your patient is still living. And that's the goal of this project. So we want to build 3D maps of the distribution in whole human brains, and use these data sets for validation of PET tracers and other neuroimaging modalities that you want to use. And use this data also for validating and understanding how the disease is affecting the brain and causing cell damage. And that's where the imaging is going to come in, and the deep learning is going to come in. So our workflow looks like this. We have patients who are enrolled to this program, and they went through MRI and PET scan in vivo. And just as a disclosure, I'm not going to be showing the PET scan images here because it's actually a project with a pharmaceutical company, so I'm not allowed to show the PETs, just the part of the project that I did. So the patients go through MRI and PET scan in vivo where they're still living. When they pass, we get the brains. And they go through fixation, and yes, we have 3D printed skulls because we do the models from the CT scan and use this as molds. And why do I do that? Because I want to reduce the deformation. So interesting fact is that the brain is very soft. It's almost like a jelly. So when the brain is removed from the skull, it squishes. It loses its 3D. It loses a bit of this original shape. So we're trying to use these molds and reduce these artifacts. When it's fixated, it takes a few weeks up to a couple of months. So it's actually a long-term process. It goes through embedding in chemical that we call celloidine that solidifies, and you get this block here that goes through cutting. During the cutting, we take images that we call block phase. So those pictures, we have a camera located just on top of the microtome, and we take a picture every before each microtome is stroke. And why we do that? Because that block phase is going to work as a template for the original brain shape, and I'm going to need this later. And after the cutting, it goes through a series of chemical stainings and embedding, and you have this slice. You end up with this glass slide here. The problem is when you do this staining and cutting, you really introduce a lot of artifacts in this tissue. This tissue is going to lose all these like original shapes, and going to have tears and holes, and it's going to be basically almost impossible to recover the original brain shape. So, and by the way, this tissue is being stained for what we call 88 and 8200. Those are markers. They stain for tau. And 8100 is going to be staining for tau that it's in the early process of phosphorylation. So what happens with the tau proteins that our brains have tau? It's a normal thing in the brain, but in Alzheimer's, for some reason, it begins to accumulate and begins to misfold and misshape. And it goes through a process that takes some time. So 8200 is going to be staining for new tau, and 88 is going to be staining for tau that is older, that is being misfolding for a longer time in the brain. So what do I need to do? I'm left with this huge slide that doesn't, by the way, doesn't fit a normal microscope. And these images are called block faces. How do I go from this material to neuroimaging? And why do a direct comparison? how can I do direct comparison between the information I get from those images and some neuroimaging modality? And there was this project, and we had to come up with a whole pipeline, and where we had to develop the imaging equipment and the software for the equipment, all the pipelines for stitching, doing the segmentation, pre-processing. And in the heart of this pipeline, there is a convolutional neural network that does all the protein localization and segmentation. So the first thing was developing an imaging platform for those slides. As I said, the slide's too big, it's 6 by 5 inches. And we don't fit a regular microscope. The microscope stages are way smaller than that. So we developed an in-house scanner, which is a prototype. It doesn't look that glorious, but it works well. It has scanned more than 500 slides by now. We also developed the software. And it was a very cost-effective for the lab. So it way cheaper than a commercial scanner. So the way it works, we scan, it's about 1.22 micrometers. It's about 0.5x magnification. It's not a lot of magnification for microscopy, but it's enough to see the protein. And for each slide, we can have about 20 gigabytes of raw images for each slide. And by the way, each brain has about 800 slides, so each brain is going to be about 20 terabytes. So it really depends on the size of the tissue. Not all brains have the same number of slides, but it's going to be something around 20 terabytes of raw data. So these tiles that are scanned, they go through stitching process. And by the way, most of this pipeline runs on a cluster at UCSF. And during the stitching that might take 48 hours, we end up with another 20 gigabytes of image. So we have another volume of 20 gigabytes for each slide. And that's how they look like after stitching. And inside these huge images, they're going to have something around 50,000 by 80,000 pixels. You want to locate these tiny black blobs. I don't know if you guys can see it. And those blobs are the tau proteins that you're looking for, the protein inclusions. So it's obviously that you need to automate this. You cannot just have someone on Photoshop drawing masks. And that's where the imaging techniques came. So the first thing that we need to do in this project was pre-processing the block face images. Remember, the block face are those pictures that we take of the block during the cutting. And that's how they look like. And I need to segment and get rid of the background on all of the 800-something images. So for that, we use use classical image segmentation techniques. The first thing that we do is converting those images to LEB space. And in this space, you can do some thresholding. It's really easy to threshold and get rid of most of this dark background here. But the problem is usually you're going to have some structures that are still left, like the blade and this piece of the floor here. So for that, we have an algorithm that refines the segmentation, and it's based on graph theory, where each element of the segmented image is going to be node. And they're connected by edges, the weighted edges. And the weights are given by an equation that takes into account the color, similarity in color, and the distance. And when you have this graph, you can just use one technique called any cut to separate the graph. And usually you're just left with the brain region. That's how you do automatic segmentation. That's pretty fast. And semi-automatic, the user just had to select some pixels on the background and some pixels on the brain once, and you can pre-process the whole volume. So once you do the segmentation, you just stack them, and you get your brain shape back together, right? No, not really. Sometimes what happens is that when you're cutting, the block moves. So you're going to end up with just like kind of jagged volume here if you just stack them. So what you do is a first bit of image registration, where you're going to register all the slides to one slide in the middle. And it's called a fine registration because that's a kind of registration that only allows translations, rotations, rescaling, and shearing. So it's a basic, simple kind of registration. But once you have this volume rectified, you end up with this kind of volume here, and that's the best way to get the original shape of the brain. And that's going to work as a template for my map. And that's the block. And this is, I'm spending a lot of time in this block phase because that's what allows us to do comparison between the histology and MRI. I later I can just align MRI to this block and have a spatial correlation. So the second thing was processing the histological images. One thing that when you do the stitching, you're going to create a high resolution, a full resolution histology, but you also create a downscaled resolution, downscaled image. Why is that? Because usually when you try to the full resolution images that have 20 gigabytes each, you run out of memory, and it's also very time consuming. So some of the preprocessing is performed in the low resolution image, and that's how they look like. Some of them really broken. So it's really hard to align them to the block face, but what you need to do is basically align them. They have to be aligned to the block face. Some of them are really broken, and this is a really extreme case. Probably this one was, we just like threw that away. We didn't use this one. But what you do is a mix of in-vivo registration. So for those who don't know, I'm saying image registration. What I mean by registration is those are a set of imaging algorithms. They're made for aligning images. And it sounds like it is a problem, but in terms of computation, it's actually pretty complex. Because we always end up in a kind of you always end up in an optimization problem, and which is not convex. So you have all this ways you have to find a model that works for your problem. You have to find a set of parameters that deform well the image. But here I'm doing a very simple thing of just using some landmarks. So you have a user selecting points on the image, align them to the blocks. And you do this to all the images. And by then you're going to end up with something like this, which is this is the histology aligned to the block. And why am I doing this? So now that I know where those histological images go inside the brain, when I compute my maps, I know exactly where to put them inside this 3D volume. That's how I built my three-dimensional protein map. So I save these registrations for later, and I move on to the next, and probably the most interesting part of this pipeline, which is the actual deep learning. And the first thing you want to do, so the actual segmentation of those proteins is performed using a convolutional neural network. And the first thing that we had to do in order to have it working was creating the training. So we randomly draw. So we randomly draw patches of image from all those histological slides. Remember, this is running on a cluster, so I can just run it in parallel for each slide. So I randomly get patches of images from weight matter, which is this region here, and save them. And from this big data set, I select 100 for each marker. And from those 100, I create labels. I manually create labels. And the things that I'm labeling are kind of hard to see. I don't know if you guys can see it here, the cell bodies. This red arrow is pointing to a cell body. We have another thing that we call thread, which are like proteins. We call threads the proteins that are in the exons. Then dendrites are just outside of the cell. And we have other things that we call plaques. And our initial idea was to have a multi-label, multi-class classification. Because, why? Because you see, in some of the PET tracers, they bind better for cell bodies, in cell bodies. The other tracers, they bind better for axons and other kinds of tau. So this would be relevant to understanding what the PET tracer is doing in the brain. Well, in our trainings and our experiments, it didn't work well, so we just started doing a binary. For now, we're just doing a binary classification, but that's the kind of label that we have. That's manually performed. We used Fiji plugin for performing this labeling, where you can, it's a trainable plugin where you just select a few pixels from each class. You train a linear support vector machine, and it creates initial mask that you can refine later using something like Photoshop or GIMP. And well, we did this for all the patches, and those masks is also important for doing the validation of the network later. So that's the network that I'm using. That's a UNet-based network, but that's not the original UNet that was published in 2015. That's one version that was designed to work specifically with the 1 square millimeter, 1 square millimeter of tissue from the scanner, and it's also made to work with RGB information. And it was developed in TensorFlow and carries in TensorFlow. So for training, I'm adjusting the learning rate using the cyclic learning rate trick. And once you have like the initial estimate of the learning rate, we begin training. We do extensive use of dot augmentation. We do the regular rotations, flipping, shearing, and we let it train until the loss value plateau, which usually take around 2 days. Okay, so how the actual segmentation works. For each big slide that we have that has those like 20 gigabytes of data, they are broken down in smaller tiles on the cluster. All the tiles that have just background or just white matter are eliminated, so we just keep the gray matter slides. And those go through the segmentation. And this network actually outputs a probability map that can be thresholded into a binary mask. And we do this for all the tiles that were created from all the brain slides. And we recombine them together, and at the end, what you have is a huge binary mask that is the same resolution of the initial histology slide. And from that, we can compute the quantitative maps of tau. So we quantify the average amount, the percentage of tau that you have inside 0.1 square millimeters of tissue. And we do this for all slides, and we create what we call heat maps. And that's how they look like here. A better visualization. You can actually see, so here it's the heat map, and here is like the hippocampus from where this heat map was computed. For comparison, you can see that you can actually see the layers of the hippocampus are visible, and here the hotter the color, the denser the tau. And once you put them together, and that's where the 2D registration came in. So that registration that you did in the beginning, where you aligned each tissue, each slide to the block face, now we can apply those registrations here, and you have these maps inside the brain space. And once you have them in the brain space, you can do what we call 3D registration with the PET scan and MRI. And we're using what we call differential registration, PET scan registration, which is an algorithm that is really good at handling really big deformations. And we end up, that's an example of a heat map on a block face. But you end up with like the 3D volumes. And from the 3D the volumes, you can do visualization, interesting visualizations, like this one. That's a reconstruction of the protein map on top of the brain block. And on the left, you have a patient who was actually considered control. So it was a person that didn't have any symptoms. But when we went through the histological process, you realized that it was actually Alzheimer's patient. And you only see tau protein and the hippocampus, which is what's described in medical literature. Here is a late stage patient. And you can see how you have way more tau. And by the way, like the brighter the color here, the higher the density of tau. You can see that you have tau across the whole brain. And just out of curiosity, I have some videos. This is reconstruction of the early stage patient. And the purple blobs are the tau accumulations. And that's the late stage patient. And you can see have way more protein as expected. Okay, so, but now that I have this network working, how can I be sure that it's giving me a reliable result? It's giving me a reliable maps. So the first thing that we did was plotting the rock curves and computing the area under the curve for the networks that are trained for segmenting both 8100 and 88 were the two markers that I'm using in this project. And so the way this network was trained, we had 3 data sets. We had a testing data set, a training data set, a testing data set, and a validation. And the validation was never presented to the network during training. We just kept fine-tuning. We just kept using training, testing, fine-tuning the hyperparameter. And at the end, when everything was working, we ran the network with this validation data set. And here, these curves, like the purple curve is for the validation data set, and the orange for the testing. We are getting areas under the curve of more than 0.8, which is a good result. Not state of the art, but it's a good result. Maybe if we have better images and more training data, we can improve it. And we also analyze the precision recall curves. So here, this is like the red stars, they show where is my minimum threshold, the threshold that I use for creating the brain masks, the binary masks from the probability maps, and which I'm using a 0.77 value, which is a arbitrary value that we chose, and you can see that it's giving me high precision but very low recall. But I think it's actually a good thing, because I wanted this system to have high precision so that it gives me, even though it's missing some tau proteins or some pixels are classified as tau, I know that what's giving me as tau, it's a reliable answer. Because that's the kind of thing that I want to have for validation of a product from a pharmaceutical company. So everything that is classified as tau is very likely to be tau. But I wasn't very satisfied with just the rock and precision recall curves, so I decided to play a little bit with network inspection techniques. And the first thing was trying to use the class activation maps, gradient class activation maps, and how do these maps work. So from regular networks like AlexNet, where your network basically outputs one class for your input image. You compute this by multiplying the output of your last layer with the gradient of between the last layer and some other layer that shows inside the network. And basically, the gradient is gonna kind of give you the pixels of the image, the pixels of the convolutional layer, like the one that I'm using here. They're more important for the classification So what I did, the problem is this network doesn't work with a single output, class output. It actually outputs to the map. So we had to do some adaptation. And basically what I did was, instead of selecting one single pixel from the last layer, I had to select pixels. They would belong to a specific class. So this red dot here are the pixels that I manually selected that I knew that should be tau. And I computed the CAN map for all of them and computed the average. And I don't know if you can see here, that's my class activation map for those pixels. And you can see it has good localization with what would be a cell. And also I did the same for the background. And you can see that you have activation, which is basically for background. But another experiment that I did for testing if this network was really reliable was just adding artificial pixels to the image. So I covered one of the cells with a patch of image that was artificial, was manually created, and it's indicated by this arrow. And I ran a segmentation again to see if the network would be, what kind of output the network would give. And I also ran the CAM for the foreground and background. And those are the results. You can see that the network is no longer recognizing this as a cell. It used to be a cell here, that's the segmentation. It's also the class activation is no longer finding any interesting feature, whereas the patches, the foreground class activation. But the background class activation say it's background, and that's correct because that's a patch from the background that I just used to cover that cell. So I did the same for other kinds of tau that I find in those images. So this figure up here shows a cell body. Down here we show what's called a plaque, and that's another way that the tau presents itself in the brain. And that's the segmentation that we get. So is also visually looking good. And that's the addition of the artificial patch of image and the results of the segmentation. So I did this to another cell body, example of cell body. This one is a little bit out of focus, so the results not so interesting. Now, last note, it's about the dice versus label quality versus resolution. So although I was having a good classification, like the raw curves were good, the precision recall was good, the CAMs were showing the relevant result, I was still getting a really bad dice, and I don't know if you know the dice coefficient. So the dice coefficient tells you how well located one image is, say one binary mask is in relation to the other one. And it's a value between 0 and 1, and the higher the value, the better your colocalization. And I was getting really bad dice. I was getting dice that was like 0.1, 0.3. So I went to check my images, and I was like, why is this going on? Because my classification seems to be working well. And when I began manually checking the images, I realized that, so these images here, the red outlines are the labels, the manual labels, like my ground truth. And the green ones are the network segmentations. And actually the network is missing seems to be missing a lot of things. But when I visually inspect the images, I actually don't see almost don't see anything on those images. You know, like this was labeled tau, but I barely see anything on those images. So that's probably a case of mislabeling. Although these labels were performed by people who were trained in histopathology, and they were double-checked by histologists. Same with these images down here, where you see a little bit of, this case here, you see a thread, but it's kind of out of focus. So the network also has a really hard time picking things that are out of focus. So that's open question, that how we're going to improve these results right now. Maybe if I lower my threshold a little bit, but I still want to keep my precision, so my high precision, so maybe that's not a good idea. question of image quality and resolution. I don't think this scanner has enough image quality and it's a high resolution enough to show maybe threads. So this is an open question. How do you balance between image quality, really following, really having a result that is compatible with the ground truth? How do you know your ground truth is right? Because this ground truth was supposed to be right. It was done by professionals, but you still see that you have mislabeling here. Yeah, so that's a work in progress, trying to work on these questions and improve this segmentation quality. And so once we have this, almost ready, but once we have this data here, what are the things that we can do with this? That's the big question. So this project is about creating this ground truth data. And now that we have these maps, we can begin asking questions. The first thing is like, are those tracers working correctly? So we can begin doing spatial comparisons between tau signal, PET tau signal, and the heat maps that we have. We can also begin asking questions like, how is this protein distributed across the brain according to brain regions? Does the distribution follow? We know that the tau distribution follows specific paths in the brain as the disease gets worse, but we can begin to quantify how much tau you have in one region of the brain in comparison to the other one. So you can begin to ask these more like data science-like questions using this data, this raw data. One example is, do I have in late-stage patients, do I have more protein in what cortex, part of the cortex, do I have more protein? Does it correlate with the symptoms that the patient had? And you can begin to do this kind of validations using this kind of data. And this is a very general method. We are using this for tau, but this can be retrained for another kind of protein and adapted. This kind of method can be adapted to other kinds of images as well, histological images. And yeah, so thank you. That's my current project at UCSF. And
Do you have any questions? Hey, nice talk. I have a question because I'm a beginner of learning. You mentioned that you see two trends, like 8% regulation, 10% for another, 8% for
yeah, I human labeled first, and to validate the network result, I compared the human labeled images to the network output, and that's where that's how you plot curves. That's where you know what's the false negative, but then it was created by network, the binary method was created by network. So I'm assuming my human was and yeah, this validation. The ground truth, the human label, the human performance labeling. I don't have another way to know what's. Between patients, yeah. Between brain regions, you also have this change, yeah. That's one of the things that you can begin quantifying using this kind of math. You can quantify how much tau protein you're going to have in one, say, the hippocampus and other regions of the brain. It's kind of known. That is not a completely a new question. This has been being mapped over the years by histologists. So you know that the tau begins to accumulate in the hippocampus, and then it goes to other regions of the brain. But right now we just have 2 patients. So the idea is that you have more samples over time, but it's just that it's kind of complicated to get that material.
Hi, thank you. I was just wondering, can you go over again what was the basic input just a portion of one block, or can you explain that again?
Sure. So each slide, is it still? So for each brain slide, let me show you here. Like this one on top here. You break them down, and they have some pre-processing where we remove the background and white matter. You break these images. Those images are pretty big, into smaller tiles. Because if you try to work with them like this, you're just going to run out of memory. So each smaller tile goes through the segmentation process. So we have, imagine you just broke these smaller squares. Each smaller square is fed into the network. Actually, the smaller squares are broken into smaller tiles with overlapping, because the network inputs just 204 by 204. And those big tiles are something around like 1,024 by 1,024. And you each small tile, and you create the binary matches, and you can keep everything together back to a big image. And then when you have the squares get segmented, you can stitch it back into a bigger image. They're always broken down into. Yeah, we needed to treat structure because I want to do a spatial comparison with other imaging modalities, like MRI or like PET scan. So I need to know where this, for instance, this hippocampus is inside that MRI. And that's why you do 3D registration. So the 3D registration deforms the MRI, deforms the block to fit into the MRI. And then you could do direct comparison.
3D information after in training the model?
No, it's just pixel level training. Yeah. Yeah, so here what you do is like you do a first segmentation using just thresholding in the OAB space, and that's going to get rid of most of these dark regions here, which is the background, the block. But you will end up having other things that are left behind, like usually like this glare and the pieces of the blade, the floor. And a way to get rid of them without having to manually do it was you get each of the regions that are still in the image as nodes of the graph. It's a weighted graph. So, and they're all connected together. You do the connection by computing the, the Delaunay triangulation. I don't know if you heard about it. Delaunay is going to create a graph. And you populate the weight of these edges. You have an equation. I don't have it on the top of my mind. I can check the paper. But it basically takes into account the difference in color and distance between these edges. And it creates a weighted graph. And then you can begin trimming this graph, just getting rid of the weak weights, the smaller weights, and keeping the strong ones. So in general, what's going to happen is going to get rid of everything and just going to leave the nodes, the important nodes. The thing is, like here, this case here, you don't really need this. The problem is when you have one image like this, where you have the lobes and they're separated. So when you run the segmentation, they're pretty distinct regions. And I want to get rid, I want to keep them together, but I'm going to get rid of those things like the blade. So it's important to have the graph, because those three regions are going to have like a high value, while the other one's going to have a weak connection. That's how it works. Hey.
I had a question. Since your model was trained on these slides, deceased tissue culture, if you had any concerns about that translating to your eventual goal of having it be able to work on live PET scan?
So it's not tissue culture. Those are actual human brain slides that were cut. And that's what makes them complicated, because tissue culture is easier to, usually has like a very flat background. But what was the other question?
Do you have any concerns about it translating to working in a PET scan?
How you translate, what are the concerns?
Yeah, translate your model and still function based on PET.
So the PET scanner, I don't know if I'm getting the question right. So the problem with the PET scanner is like, usually those tracers, they're developed in animals, and then they're testing that tissue. And then when you go to a living human, you don't know what happens to the chemistry of the brain. You don't know what the tracer is how it's gonna react in living tissue. So we are not really modeling how the PET tracer works. We are creating maps that you can use for checking if this trace is binding to the right things. So you go to the map and see, whoa, I have a lot of protein in this hippocampus. You should see a lot of signal in this PET scan. So if you don't see it, then this PET trace is not working well. Or if you go and you see a lot of signal in some other region, there's a region called basal ganglia that has a lot of, in general, has a lot of signal on the PET traces, but when you check the maps, you don't have tau, so it's binding to something else. And that's why you need these maps. You need to validate those things or understand how they work. It's not that, you're not gonna, usually what the companies do, they don't throw the product away. They just try to understand how this product is behaving so that you can, when you do like a PET tracer on someone, you kind of know what's going on. But it's not modeling how the PET works. Okay.
Any more questions?
All right, let's thank Maryana again. Thanks, Maryana.
Thank you. Thanks.